All right, I think we will go ahead and get started. And again, welcome all. It's Mitchell Warren from AVAC, and delighted to welcome you to the next in our series of HIV Vaccine Awareness Day webinars, which I will describe in a bit more detail in a moment. But first, just some very quick logistics for today. I'm delighted to welcome all of you, and particularly Fatima Laher from South Africa, who's going to be talking to us about a trial that many of us have been waiting a very long time for, and you'll hear about why and what it all means and what it could mean in just a moment. We will be going through her presentation in muted mode, so don't be surprised that you are muted. We do welcome questions throughout, so feel free to email at any point to avac@avac.org, or if you're on the Ready Talk feature and watching along on the slides, you can always enter questions in the chat feature. And then as soon as the presentation is done, we'll take those questions, but also I'll give some instructions on how to open the line so people can engage in a conversation. This is the third of 5 in our series, and I will just put up the slide to show you where we are in the sequence. If you go to avac.org/HVAD, you'll be able to see the full list of these 5. And for the first 2 that have happened already, an update from the Janssen program on the adeno 26 mosaic vaccine candidate from a couple of weeks ago, and last week's history and future of the NIH's Vaccine Research Center. The recordings and slides are available at that website, as will Fatima's when this call is done. And we have another call this Thursday with Lynn Morris from South Africa, and then another one next week on HIV Vaccine Awareness Day itself that wraps it all up next Thursday. And you can register for those on the same site, avac.org/HVAD. For those of you, if you weren't on last week's call, you can certainly see Barney Graham's presentation from the Vaccine Research Center. But one of the things he talked about was the history that leads us to May 18th, and he did a beautiful job of explaining that, a very important speech on May 18th, 1997, and that's why we've had so much focus on these calls. If one looks back on those 20 years, one of the most exciting and important moments was actually in 2009, which seems a very long time ago, when results came from a very large trial in Thailand that you'll hear a bit about today. And you may wonder, what does the result in Thailand have to do with a major trial in South Africa? And I'm delighted to hear about that journey from Thailand to South Africa and beyond, hopefully. I'm delighted to welcome Fatima Laher to the call today. She is, as you see, based in South Africa and is the protocol co-chair of the HIV Vaccine Trial Network trial number 702, and you'll hear about 702 in just a moment. And she is the director of vaccine research at the Perinatal HIV Research Unit in South Africa, which has really been pioneering research not only in vaccines but across HIV and AIDS for many, many years, founded originally by Glenda Gray and James McIntyre. But delighted to welcome Fatima here today to provide an update on HVTN 702. And with that, I will turn it over to you, Fatima.
Thank you very much, and thank you very much for having me. This is a very special cause in my heart. It is the journey of hope to prevent HIV. And people will look at the screen and see, what is Uhambo? Uhambo means the journey. And it was community representatives that chose this slogan to represent what it is they felt in their hearts was required for this region, Sub-Saharan Africa, that is so incredibly affected by the HIV epidemic. So Uhambo is the whole journey, and we're going to talk about the whole journey today. But HVTN 702 is something very specific that we're going to offer updates on in this webinar. HVTN 702 is the first HIV vaccine efficacy trial, which is happening now in South Africa, to be done in 7 years. So it is, no matter the results of the study, it is a landmark study in HIV vaccine trials. I'll move us on to the next slide. Sorry, Mitchell, am I able to move slides? Great. So this group, this audience does not really need to be told about the fact that HIV is not over. It also does not need to be told that we have made tremendous efforts so far to treat this epidemic in a humane way and to make inroads into HIV prevention. But we are going to need every prevention technology we can get to end this epidemic. If you look back at the turn of the millennium in 2000, there were 3.2 million new HIV infections that year. By 2015, there were still 2.1 million new infections happening. So we haven't reduced enough, but we've reduced some. We had over 1 million people on HEART in the year 2000, and now 17 million people are on HEART, which is an incredible victory for the field. Unfortunately, in 2015, 36.7 million people were living with HIV, and that means that if you take 36 minus 17, the remainder of people do not have access to HEART. We cannot keep up with the number of people that require treatment, and this is why there continue to be AIDS-related deaths. There were 1.5 million in the year 2000 and another 1.1 million in the year 2015. Because of this, we can say without a shadow of a doubt that we will need every prevention technology that we can master to roll out into the population so that we can stop new infections and rescue our healthcare systems. Nowhere is this more apparent than in the sub-Saharan African region. I'm going to move us over to the next slide. This is a very cramped slide, but it tells you the journey so far of what has happened to make the successes in HIV vaccines possible. It started in 2009 when in Thailand they announced the results of the RV144 study. It showed that there was a 31% vaccine efficacy. This means that when comparing the vaccine arm with the arm that got nothing, that got placebo, there was a 31% difference or reduction in HIV acquisitions. And this was using 2 vaccines. This had not been done before. Before, we had been using a single vaccine on its own and trialing them out in isolation, a practice that to date had not worked. But by 2009, this combination approach using a vaccine that was called ALVAC and another vaccine that was called AIDSVAX, made for subtypes B and E of HIV, with an adjuvant called alum, showed this remarkable vaccine efficacy. It was remarkable because it was the first vaccine ever to prevent people from getting HIV. And it showed us that a preventative HIV vaccine is possible. As time went on, and as the results were studied even further, it showed us the scientific principles behind what the protection through the vaccine was pinned behind. So you will remember that in the old days, people thought that getting a neutralizing antibody response to a vaccine would be what would cause protection against HIV acquisition. But as it turned out in the Thai trial, this was not how protection was mediated. Protection was mediated through something called a binding antibody. You'll hear this quite often, these IgG type of antibodies that we see against a part of the HIV virus, which is the envelope of the virus, and a very specific part of that envelope, which is the V1, V2 loop. Knowing this has made really important advances possible. The downside of the ALVAC and AIDSVAX BE regimen was that the protection and the immune response did not last for a very long time. And what they saw was that by a year after completing the vaccine, the immune response had dropped quite low, and so too did the efficacy being the actual protection that it provided against HIV infection. So in 2013, we did a study in South Africa to see if the same vaccine regimen would provide immune responses in South Africans. And that study was HVTN-097. We did find that the Thai regimen had a very good safety profile in South Africans, and it also had a slightly better immune response in South Africans when compared to those in Thailand. And this can be ascribed probably to regional differences, genetic differences. We then moved on to HVTN-100, which was a very exciting study that started in 2015. And what it did was that it adapted the vaccines that had been used in the Thai study and in 097, but it adapted it for the virus that is circulating in the sub-Saharan African region, so subtype C. And so it took a subtype C of LVAC and a bivalent subtype C GP120 and combined it with an adjuvant called MF59. A phase 1 and 2A study was performed of these products, and we're very happy to say that it had good safety and very good immune responses as well. At this time, we are currently analyzing if giving a booster vaccine at month 12, that same time point at which the RV144 vaccine responses began to wane. So in HVTN-100, when we gave the month 12 booster, was it able to prolong the immune responses? And while that is happening, HVTN-702 has started. It started in 2016. It is using these same subtype C vaccines in a phase 2B and 3 trial. That means that beyond safety and immune responses, we are now looking also at efficacy. Does it really prevent people from getting HIV? That is what is called efficacy. And it is so exciting to us because it is possible that this could lead to the world's first licensed preventative HIV vaccine. We don't know yet. That is why we are doing the study to find out the answer. But if it does happen, then this could lead towards that effort. Let's move to the next slide. Very briefly, you will be able to see the time points at which we are doing the HVTN-702 vaccinations. There are months 0 and 1 in which we use LVAC, and half the people get placebo. Months 3, 6, and 12 in which both LVAC and bivalent subtype CGP120 MF59 is used, and half get placebo. This is what is called a randomized, double-blind, placebo-controlled clinical trial. It means that neither the doctors nor the participants know what that participant individually is getting, and half are getting placebo and half are getting vaccine. And everyone will know at the end of the study, when everyone is unblinded, what they really got. Let's move to the next slide. So HVTN-702 is a 5,400 person trial. This is a very large trial for healthy HIV-negative men and women. They have to be aged between 18 and 35. The study opened in October 2016 at my own trial site in Soweto, Barra, where the first HVTN-702 vaccine happened. And interestingly, it was also where the first HVTN-100 vaccine happened. So the Soweto, Barra site has a very important place in vaccine history. We have had excellent retention in the whole study at all sites. So you can see 98% retained at vaccination number 2, 99% at vaccination number 3, and all so far have come for vaccination number 4. Let's move to the next slide. This is the bottom tip of Africa. You can see South Africa that is in the blue color, and you will be able to see all the 15 HVTN 702 trial sites. By the end of April 2017, we had enrolled 526 in 11 enrolling sites. There are 4 sites that are yet to enroll. They are yet to open, and you can see them in blue. But we're so happy that things have been going well with enrollments thus far. Let's go to the next slide. And very soon, I'll put you into an update. We are moving into HVTN 702 protocol version 2. Some of the sites have started transitioning, and the rest will follow suit very quickly. And some of the changes, which are welcome changes, is that we will be giving even more details on the HIV prevention package to participants, including how to access PrEP. As you know, PrEP has been a developing landscape in South Africa as we implement and open programs, and the availability of PrEP is something that is very relevant to our participants. And we're also going to be giving extra details on STI testing and treatment. We will be collecting data about PrEP use. Currently, we are taking histories about whether people are using PrEP or not. And in version 2, we will also be taking a very specific type of blood sample called a dry blood spot in order to measure whether people really are adhering to PrEP or not. And this will give us feedback on what the levels of PrEP usage are within the study. Let's move to slide 8. Of course, while 702 is going on, we're learning a lot from the data analyses of HVTN 100. In HVTN 100, the immune responses became much lower by month 12, but then when we gave the month 12 booster, the responses became higher again. So we will also boost at month 12 in HVTN 702, but we are asking ourselves, do we also need to add a month 18 booster to HVTN 702? And I'm going to ask you to watch this space, because this is another evolving landscape. As we learn more and understand more, we will be able to change the protocol more to benefit participants. Moving to slide 9. We're very pleased that HIV vaccine trials have a wonderful relationship with local communities and regional stakeholders and national stakeholders. And all of them have been really involved in this effort, and they continue to be active since the first HIV vaccine trial ever opened in the country. Our trial participants are part of our communities, and the prevention methods are being developed for communities. So it's important that all community stakeholders are involved. The stakeholders need to have opportunities to ensure that the research ties into community preferences, our needs, our customs, our traditions, and our beliefs. Some of the more recent work that has been done with stakeholders involve a workshop in the middle of last year, and this was for community educators, recruiters, and community advisory boards, and they helped develop some of the materials that you see today. And there were also community and stakeholder consultations, one in September 2016, a follow-up in March 2017, which provided a platform for national, provincial, regional, and community level stakeholders. To move to the next slide. There's also been widespread media interest we have our own media presence through the uhambo.org.za web address. And the media have also covered the trial since its inception. So going back to last year, there has been quite a lot of coverage around the vaccine trials. Some of it shows that there are people in the community who are concerned about the nature of a vaccine. They have deep distrust of what a vaccine constitutes of, and we see this in South Africa in small pockets. Most recently, it has affected the control of measles in our country, where we are starting to see measles cases break out very recently because people or select people are not taking their families in for vaccination. And measles is a part of our EPI program of vaccination. And so we see this extended toward all vaccines, and that includes HIV vaccines. But we also see other reports, for example, one that spoke to trial participants and their communities and their families and deemed them to be heroes. I personally do think that the trial participants are heroes. Their contribution into vaccine science is something that the world will never be able to repay. Their ability to join a study is time and effort that is given as a gift to the entire world. There's also been extensive television and radio coverage within the region, and you know, those are some of the logos of some of these regional and national talk shows, which have covered these this journey of trials very, very well. I'm going to ask us to go to the next slide. In our attempt to leave the informed consent information out to the entire world, we have made sure that we have put out information about the informed consent in a video format that is very easy to watch through Vimeo and YouTube. You could download it and watch it right now. And this makes sure that every single person, not only trial participants, not only researchers, not only vaccine advocates, every single person who has an interest will be able to search for the HVTN 702 informed consent video and watch it. So please pop some popcorn and take all that information in from the video. It is very wonderful to watch. I'm going to take us to the next slide. In closing, I'm going to remind us that Uhambo is a journey, and 702 is one trial in that journey. HVTN 097 and 100 preceded 702, and it is on the track to develop a vaccine that can be licensed if it works. But we also have a program of studies that will help us to understand the nature of vaccines even more. And this information is so important for future iterations and development of HIV vaccines. And there you can see a whole list of HVTN trial numbers which look at different products. Could different products in combination work better than the types of products that were used in 097 and 100? Could different adjuvants work better? And what if we took the best vaccine antigens from RV144 and from HVTN 100, and then what if we use them to boost people who already had the HVTN 097 vaccine? Would that make them have fantastic immune responses that would last a very long time. So these are the kinds of questions we're asking. All of these other studies may not get as much press time, but they are so important in order for us to complete the journey toward preventing and ending HIV. I'm going to take us to the last slide. And this is a huge thank you to everybody that you see on the slide. It's the participants who are very dear to us, the communities from which they come, our CABs who have been standing by the research community and cheering them on even when it was not easy, to our site staff who are dedicated and have had to learn a very distinct skill set in order to make these trials happen. The P5 group, which is an alliance group for the good of public health, and they are the South African Medical Research Council, the Bill and Melinda Gates Foundation, HVTN, the NIH and DAIDS, GSK and Sanofi that make the vaccines for us. The protocol teams for all the protocols that you saw listed on the previous slide, our data lab and statistics colleagues, and most importantly to you today, thank you for advocating for the end of HIV. We are very sure that this journey will continue, and we know that everyone is committed to making it happen. Thank you for having me today.
Great, Fatima. Thank you so very much for a very clear and I think I can say for everybody on the call, a very passionate presentation. So thank you so very much. To remind everybody, you can continue. A number of questions have already come in via both email and on the chat feature here at ReadyTalk. Please keep them coming. I'm going to start with a couple, and then I will offer to open the line. You can hit *7 to unmute your line. I would ask you to introduce yourself, be brief, and then hit *6 to remute so we keep the line nice and clear for the recording that will be online very shortly. A whole bunch of questions spanning a lot of different issues. But let me start with something near the end of your talk. There were a few different questions that came in from very seasoned advocates as well as some newer in the field about the role of adjuvants. So I think first some questions came in about, could you just explain what an adjuvant does, and then a question off of that is the choices from which you're making a decision to put an adjuvant into 702, a question about which one would be in that trial, and then what might happen because of some of the other studies you described on one of those last slides in terms of different adjuvants, and might different adjuvants provide different levels of protection and what that might mean for the long term. So a bunch of questions all rolled up into the adjuvant world.
Well, that is a great question. I want to explain an adjuvant as Red Bull because that is how one of my community educators explained it to me. She says that Red Bull is not going to keep you alive, but it is going to make sure that when you're awake, you have a heightened sense of awakeness. And it's very similar with vaccines. Giving someone an adjuvant is not going to make them have an immune response to HIV. It's not going to give them protection against HIV. But it is going to make sure that if you give it with a vaccine, that the immune response to the vaccine is going to be better. So we have a number of different adjuvants that are available to us. And the adjuvant that has the longest history and is used in many of the vaccinations that we take in childhood is something called alum. And the chemical name for alum is aluminium hydroxide. And that is, as you can hear from that word, it is a chemical that is put in with the vaccines in order to make the response to vaccine even better. There are other types of adjuvants. For example, HVTN 100 chose to use MF59, and we can look at other vaccine studies to try and understand what would be the differences if we gave one adjuvant versus another, but we will not be able to do it for sure until the direct comparison with this vaccine is made. So we can have a hypothesis based on previous vaccine trials with non-HIV vaccines, and that might lead us somewhere, and this led us to MA59. We were hoping that MA59 would give us better responses, but we will not be able to know for sure until we put these things head-to-head. Another adjuvant that is up and coming is something called AS01B, and you'll be able to look back at the second last slide and see that AS01B is also being tested out with these HIV vaccines to see maybe if this one would be better. So we don't know for sure what the best adjuvant would be, and this is why we're doing the studies. Will it provide different levels of vaccine efficacy? So in other words, will the protection be different? It is difficult to say at this time because we don't have human trial data. But the reason for an adjuvant is to increase the level of an immune response, and therefore hopefully that immune response would last a little bit longer. And so ultimately it may have some benefits in terms of protection.
Great. Fatima, thank you so much. It is a complex set of issues, so super helpful to have clarified that. And a question to come in just to clarify. So 702 is using MF59.
Correct.
The Thai trial had used alum, correct?
Yes.
And then these other studies, and I just put up the slide you had toward the end of the research program within the entire P5 program, where you have some of the studies comparing MF59 with some of the other adjuvants. So is the theory then that even with a positive result of 702, one would look to these other studies to compare and perhaps even change for the future vaccine delivery with a different adjuvant if one of these other studies did show better immune response? Is that a fair summary?
Absolutely. And to remind everyone, in 097, we didn't use MA59. We used alum because 097 replicated the regimen of RV144 exactly. And so all of these studies are being done in this coordinated fashion because the information that feeds out will be able to inform future development.
Great. Super helpful. Let me ask an additional question because it relates to the slide that's up on the screen. A number of people asked questions about the relationship of the products being tested in South Africa and the original products in Thailand. And one of the things you talked about, of course, was the shift in the subtype to which this vaccine is addressed. So this whole program looks at the subtype C that is so common and important to address in South Africa. But a question came up in terms of other studies about how this particular vaccine might work in other subtypes, or if there are other studies looking at, well, if it works in 702, how do we know if it would work as it is in Thailand? Or what about East Africa where we have different subtypes? So what might happen in terms of making sure this vaccine is useful in non-subtype C areas?
That is a really interesting question. What we're able to do is studies about immune responses that looks at whether people would develop something called cross-clade responses. This means, yes, of course, we expect for you to be able to get an immune response against the subtype C HIV because that is what we immunized you with, but would you be able to get a response against some of the other subtypes? So A, B, etc. And certainly what we are seeing is that this vaccine regimen, the LVAC and GP120 vaccine regimen is able to give these cross-clade immune responses, and that is quite exciting. I know that Janssen had talked about a similar adaptation of this kind of regimen to include a strain against all strains of HIV, to make a global HIV vaccine, and that is really exciting news. But we would be able to do something called bridging studies to see if results that we're getting from the lab about cross-clade responses would be able to be translated into other populations when tested anywhere else in the world. This is one of the really important reasons that we do these research/correlates program trials, the trials on the right-hand side of your screen, because through these studies we learn more about what the correlates of protection are. So what is it that you are developing in your blood that is made as a result of the vaccine so that you get protection? And once we know what those markers in the blood or correlates are, then we will be able to use that correlate to test out any other person in any other parts of the world. We will be able to test out people who are not 18 to 35 years old, so in other words, younger people, older people. We will be able to test out any other population that we hadn't tested out in this particular trial, HVTN 702, and give a result with a particular level of confidence. This is why discovery of correlates is one of the important endeavors for vaccine scientists.
Great, Fatima, so great. And I'm so glad you mentioned the issue of the populations and the potential for bridging studies, because a couple of questions came in asking about the target population. And so your slide early on talked about, I think, 18 to 35-year-olds, correct? And actually I'll just put this one up for everyone. And someone asked the question about if you could say a bit more about what is the target population for amongst these 5,400 beyond being HIV uninfected and 18 to 35. Who's actually enrolling in the trial that's enrolled so far? What can you tell us about them? And are they the people that you would most expect and want to be in this vaccine trial so far?
So as you know, every single trial has a very long list of inclusion and exclusion criteria to make sure that participation for the study participant would be reasonably safe. So we have a long list of medical criteria, and this trumps up to, are they healthy? And so far so good. Only healthy people are enrolling into the study. And then we have a list of criteria related to HIV. So people should be the type of people that are sort of general population people who have some vulnerability to the disease. And in South Africa, that is really not hard. So if you are having sex, that gives us all a degree of vulnerability to getting HIV. This is because South Africa is an incredibly prevalent area of HIV, and one cannot always really know the status of one's partner, and sometimes one's partner doesn't know their own status at the time. So sex itself is a risky endeavor. It sounds very odd to say, probably to people from the developed world where the HIV epidemic is clustered in very niche populations like IV drug users or MSM populations, while the general population is relatively less affected by HIV. That is not the case in South Africa. In South Africa, the general person who is having sex has an anxiety about susceptibility to HIV, and that is why they are being permitted into the trial. The age of 18 to 35 years old was chosen because this is where we see HIV acquisition being at its worst, statistically speaking. We've chosen both men and women. In South Africa, we have a particularly heartbreaking situation which we don't talk enough about, and even if you've heard this before, I really feel we cannot say this enough. I am a complete feminist, and I find it incredibly difficult to deal with the reality that the most susceptible people to HIV are young women. 18 to 35-year-old women are the highest and most at-risk population for HIV. Maybe people thought I was going to say something else, some other niche group, but it's 18 to 35-year-old women. And yes, we're seeing them enroll in the study because there are so few things that women can control to prevent HIV. Female condoms are a visible thing, and men don't always want to have their women use it. Male condoms, well, that is male-driven. Circumcision is male-driven, beside PrEP, a vaccine is a real chance for a woman to engage in an HIV prevention modality. And then, of course, part of being a feminist is also understanding where are men and how is their situation affecting the world. And what we're seeing in South Africa is that the slightly older males are acquiring HIV at a very high rate. We see a wonderful protective effect of youth in men, but young boys don't stay young boys forever. They have to grow up to be men, and as they become older, we see in sort of the 25 to 35-year-old, especially age group, that men have an incredibly high risk of HIV acquisition. So yes, these are the populations that are enrolling in the study in South Africa, and those are the reasons why we think it is so important to have those eligibility criteria. It is because we have what we call a generalized epidemic. It has hit us so hard.
Yeah, Fatima, thank you so much, and so helpful for that specific context. And particularly, a number of questions actually came in about oral PrEP, perhaps not surprisingly. As you know, many of the advocates on the line were pushing very hard for both PrEP access generally in South Africa as well as for an inclusion into the 702 trial. So a lot of people commented in emails and chat about happy to see the version 2 of the protocol that you described as being more positive toward PrEP. And so a couple of questions here about PrEP. One, could you give us some details of how it's being incorporated in terms of is it linking specifically to national programs or demonstration projects? So just details of what that looks like, how the trial team will monitor for this. And then someone else asked a question, how can you even assess efficacy with participants if they take a lot of PrEP? So what's the way in which the PrEP users will be monitored so that you get a clear answer, if you can, about vaccine efficacy compared to perhaps PrEP efficacy?
We have a particularly fortunate time in South Africa right now in which our government is really responsive to emerging findings that have public health benefits. As you know, it wasn't always this way, and that is why I'm mentioning this. They have fully supported all of the HIV prevention modalities. For example, we have tremendous national circumcision programs with incredibly high uptake. And I think part of this is because there is also a cultural underpinning that makes circumcision very acceptable to our South African culture. It has also adopted PrEP by licensing Truvada for oral PrEP as daily use, and began the hard work of developing policy guidelines and sponsoring demonstration projects and opening programs that are very encouraging to see happen. And this is happening incredibly rapidly, and with a great deal of support and enthusiasm from our national stakeholder. We also have, obviously, very passionate researchers who in some way are affiliated with or are part of these projects, these demonstration projects and programs, and it makes running a trial like 702 in the context of PrEP very easy. So as these projects are now opening or opened, the informed consent had to be updated in the new protocol version to give people this information that PrEP, in conjunction with other things, circumcision, condoms, needle exchange programs, etc., PEP, all of these things are still available, and this is the news on PrEP. So that is a very exciting thing for us. Having more modalities for HIV prevention is a plus. It is the reason why I showed the first slide. I myself continue to be incredibly traumatized by the number of people who die of HIV. And for us to be able to prevent this disease with whatever modality is available and with whatever modality people want to use is definitely part of our program. For us to be able to include in our trials access to condoms, circumcision, PrEP, and whatever new modality comes out is part of our researchers and our communities and our participants' absolute commitment to ending the epidemic. But as I had said to you, we don't have a situation yet in which we have ended HIV. We have a situation where, for reasons that are very contextual, very biobehavioral, we continue to have HIV infections, and we can only add help to prevention with a vaccine. That's what we think or we hope. And so we will still be able to tell the difference between the arms by measuring PrEP. And people have asked, how are you going to measure PrEP? We certainly are going to ask people about their PrEP uptake and take a history to say, can you show us your pill bottles or your scripts, or give us a verbal account of your use of PrEP, and we're going to note that down, like we note down all other medications participants are taking, and then we're going to test people for adherence to PrEP through a blood test, and they will be aware of this because they will sign consent for this in the new protocol version, so they should be comfortable for this procedure to happen, to be in protocol version 2. And then, we will have 5,400 participants, where I'm assuming between placebo and vaccine arm, there will probably be similar, because it is random, a similar distribution of people that use all types of prevention modalities: condom, circumcision, PrEP, PEP. And if there is a difference between the vaccine arm and the placebo arm, we will know that the vaccine arm has an advantage over the standard of care arm. When we say placebo arm, we really mean the standard of care arm, and that is, everyone having access to all the modalities that are available. So that is the plan. I agree that if PrEP uptake was perfect, if it was 100% and everyone was 100% adherent all the time and taking it all the time, you actually would not be able to see the difference. But in reality, we know that is unlikely to happen. When we look at results like the FACS results, we see that it doesn't always happen that people take up every prevention modality and is adherent to that modality perfectly. This is why HIV acquisitions unfortunately continue to happen. And in my own opinion, I think part of that is because prevention modalities that rely on adherence also rely on a person's self-perception of risk. And in a generalized epidemic, the perception of risk does not always equal the true risk. And I think that this is what the virus kind of depends on to spread as a generalized epidemic.
I think it's so very helpful and so true. If you don't think you're at risk, you won't take your pill every day, and you probably won't go get your vaccination either. So a critical issue in terms of helping people better perceive actual risk. So a number, perhaps because it's nearing Vaccine Awareness Day, a bunch of questions came in from a lot of different people that cluster around two questions that have dogged this field, and I think every vaccine advocate and researcher has faced them at some point. How good does it have to be, and when are we going to get it? So a number of questions about what would it take, do you think, to get licensed? The 31% in RV144 in Thailand wasn't deemed good enough. And so a couple of questions came in about what is your sense from the regulators and or just the mathematical modeling that has been done in terms of how good would it need to be available in South Africa beyond research? And the other related question is the timing. You've described in a remarkable journey, one that has many different pathways to it, but a sense of with 702, what is the timing, and particularly if it quote-unquote works, back to the first question, what would be the next steps in terms of when we might see either demonstration projects, licensure, or initial rollout? I know very hard questions that every advocate gets, and every advocate hates to have to answer, but there you have it. So how good and when?
So interestingly, that answer does not depend on me. I can give you a whole lot of answers about doing the trials. But those answers depend mostly on advocates moving the stakeholders, the national stakeholders. So the South African government obviously has been really engaged in this effort for many years now, initially through the South African AIDS Vaccine Initiative, SAVI, through the MRC, and through the Department of Health. And they have responded to say that when we have results of the vaccine trial, HVTN 702, the data will be put together in what they call an investment case to be able to see how much will this help the country at that time point and for how much money. So to me, it sounds very much like a cost-benefit analysis. And then the government will make a decision on whether it will roll out a program for vaccination. People want to know very concrete numbers, but I think administrations change, government administrations change, priorities in funding and in government budgets change, and this is where advocates can get involved to be able to understand the trial results when they come in 5 years' time. We don't have them right now. And to be able to understand more on the investment case at that time to either move government in a direction in which it should implement that vaccine trial because it will be of, that vaccine because it will be of huge benefit to the population, or to move the government away from investing the people's money in something that would have little to no benefit on a public health scale. I think many people have seen a common slide that is taken from a paper called The Impact of an AIDS Vaccine in Developing Countries, a New Model and Initial Results. It was published in the journal Health Affairs, and it shows that if we have a 30% efficacious vaccine, like the one that RV144 has now, with 20% coverage, that means that 1 in 5 people would be vaccinated. Then we would be able to avert 5.5 million infections, in a 15-year period between 2015 and 2030. And that was for the epidemic as it was then. If we move up to a 70% efficacious vaccine, that's now really on the high end, and if we're very ambitious, and if we cover 40% of people, then we can avert 28 million people in being HIV infected in 15 years. I think what we're hoping for with HVTN 100 is maybe somewhere in the middle. We don't know what the efficacy will be, but I think all of us would like to think that it might be somewhere in the middle, maybe 50% efficacy. It's hard to say, and if one had 30% coverage, so 1 in 3 people were vaccinated with a 50% efficacious vaccine, we would get 17 million new infections averted. I'm a clinical doctor, Mitchell. For me, every life counts. But there are people that need to make decisions for entire populations, and that time is coming soon. The years go and slip by quicker than we think, and so people advocates especially, will need to keep up with this information and understand the trial results when they come out. We will know them fully in less than 5 years, and start forming plans with that knowledge about how we engage governments.
Now, it's a great ending there, Fatima, in terms of understanding. And you said over the next 5 years. I think the one big message I think many of us got back in 2009 with RV144 and then subsequently with the oral PrEP studies is it's not too soon to plan for that success, of course, and I know you've described that well, in that with 5 years to run on a trial, we want to be able to act on information as it comes. I know we're at the top of the hour. A number of additional questions came in. I wonder if I might just quickly get you a couple of quick ones. One, and actually relates to this issue of what's happening at the community level. So you so nicely described the way in which the trial is working around a number of mechanisms through community engagement. And someone asked a question about kind of the complexity at the community level, given that many of these very same communities where 702 is taking place have not only long histories in vaccine research, but are at the forefront of oral prep rollout. Some of these sites and communities are also engaged with the antibody-mediated prevention study. Some of these same communities are aware that in the next few months a long-acting injectable PrEP agent is going to start in a clinical trial in South and Southern Africa. And you, of course, have 702. So how is all of that playing out, and are you all doing anything differently with 702 than perhaps in previous trials where there might not have been so much, I hesitate to say competition, but so much activity happening at the community level?
Well, it is really wonderful to have all this activity because it shows the entire community that we're all committed to finding solutions, and if one thing doesn't work, we will go forth and investigate 10 others until we find things that work for our communities. But we have to do things differently, of course. So one on the ground thing, for example, is that we have to have systems that prevent co-enrollment, and that's for the safety of the participants, not to be enrolled in 2 trials at once. And so, Mitchell, there is a community in Cape Town that is being enrolled in a community-wide HIV prevention trial, and they're going to have to make sure that the people that they enroll are not individuals who are enrolled in that particular study. So on the ground, it means that we have to do things a little bit differently. At team leadership levels, it means that different protocol teams have to speak with each other about where research is happening. And we also need to make, on a network level, we have to make commitments to developing trial sites in places that don't usually host trials. We do have a lot on our list, a lot of sites on our list that do trials very commonly. But we are developing new trial sites. For example, Mthatha is one of them. Places that are a lot more rural and isolated, but still have incredibly high rates of HIV acquisition. So yes, it affects us across the board, but we can only hope that it will affect communities in a very good way.
No, that is super helpful. Thank you so much, Fatima. I know we've gone a little over the time, but as you can hear, questions were coming in and all sorts of important issues, and really want to thank you and know that it's much like no trial does answers everything, no one webinar answers it all, but you've set this up so nicely right in the middle of this 5-part series that people will see on the screen. Fatima mentioned actually in her talk not only about 702, but the work around a mosaic vaccine that might work across multiple clades. That was back on April 28th with the Janssen team. Barney Graham last week talking about the history and the future of the Vaccine Research Center. And later on Thursday this week, Plan BNAB with Lynn Morris, also from South Africa, looking at antibody research in detail. And then finally next Thursday, Nelson Michael from the Military HIV Research Program, which of course led RV144, really providing an overview of vaccine development. So Fatima, your presentation right in the middle of these 5 really was perfectly timed to help us understand where we've come from with RV144 and where you all are going with HVTN 702. And I want to thank you so very much. All of you, if you missed any part of it, the slides and recording will be online later today. You can register for the other 2, and there are a whole range of Vaccine Awareness Day materials, and as we always say, DayVac HIV Vaccine Awareness Day isn't really just May 18th. It should be always, and those materials are available for download. Additional materials will be made available later this week and early next week to really help put this search for an HIV vaccine both in the context of the larger HIV response, as well as to provide updates and perspectives on not only where we are, but what else needs to happen and how we as advocates can help make that so. So thank you so very much, Fatima. Thank you all for joining, and we look forward to many more conversations.
Thank you. Thanks to the advocates and see you in 5 years' time.
All the very best. Cheers, everyone.
Bye.